Welcome again, students. So we are going to discuss with this topic, that is the DNA replication, its second part. So yesterday we discussed about this types of replications, DNA replications, then Nelson and stack experiment. So today we are going to discuss about its next part, that is the prokaryotic DNA replications. Okay. So there are the components required for the prokaryotic DNA replications. The various templates of various we can say components are there. The first, that is the template DNA. So the parent sequence of nucleotides to be used as information in the synthesis of the complementary strands. So a template is necessary as the new DNA strands is being synthesized. So each new nucleotide to be added is matching to the nucleotide on the template standard based on the charge of base pairing rules. So we can say it is then the sequence of the nucleotides of the parent DNA strand that determines the sequences of nucleotides of the new DNA strands. Okay, so the next, that is the origin of replications. So in this initiations of DNA synthesis does not begin just anywhere along with the molecules, we can say. Then the rather replications begins at the specific locations, or we can say sites, referred to as the replication origins. So the prokaryote has a single origin site referred to as a, we can say, ORIC, ORIC C. At these sites the initiations of the replications occurs. So in eukaryotic cells employ multiple sites on each chromosomes that acts as a, we can say, origin for the initiation of DNA replication. So these sites are referred to as the origin of DNA replication, that is ORIS. So next, that is the opening of DNA at the ORIC site, ORIC side. So the DNA A, DNA B and the DNA C are noted, we can say these are needed to recognize the origin and to separate the strands of the parent DNA. So helicase enzymes now binds to single stranded DNA and helps unwinding double stranded DNA throughout the process of the replication. So single stranded binding protein, that is SSB, also bind to the open single stranded DNA to avoid rewinding of the parent DNA. Okay, so in the next, that is the nucleotides. So the synthesis of DNA requires for nucleotides is the deoxyribose triphosphate form and 4 in the ribose we can say triphosphate form. Okay, so the nucleoside needed for DNA synthesis are deoxyadenine 5, like we can say 5 in triphosphate, that is a DATP, then the deoxyguanosine, that is a 5 triphosphate we can say DGTP. Okay, and the third, that is a deoxycytidine, that is a 5N triphosphate, that's a DCTP, and the next, that is a deoxythymine, that is a 5N triphosphate DTTP. Okay, then the nucleotides needed for the synthesis of the RNA primers we can say are adenosines, that is ATP we can say, then guanosines, that is a GTP, then cytidines, that is a CTP, and uridine, that is a UTP form. Okay, so there are various enzymes also needed. So many enzymes are needed for the replication. In this, we can say DNA polymerase 1, which removes the RNA primers and replace them with the DNA. So it is also used in DNA repair systems. Okay, so DNA polymerase we can say so DNA polymer first has in fact 3 enzymes activities, we can say. So DNA polymerization, exonuclease activities, second, that is exonuclease activity in a 5' to the 3' direction, used for the removal of the DNA primer. And the next, that is exonuclease activity in a 3' to the 5' directions. So in this we can say proofreading activity Okay, then the next, that is a DNA ligase enzyme. So we can say it joins to the Okazaki fragment by making phosphodigester bond in between two DNA fragments. So the DNA ligase are responsible for, we can say, connecting segments during the replication, repair and recombination. So all ligases join we can say a 5-end phosphopyrrole group and 3-end phosphopyrrole group on adjacent fragments, so thereby sealing the nick. The reaction, we can say, occurs in discrete steps and requires an energy sources. So it may be in the either ATP, which is in the eukaryotic one, or we can say NAD, that is in the prokaryotic ones. Okay, so in this DNA replications, so the parent strand of the DNA, which runs in the 3-end to 5-end directions towards the fork, and it is able to be replicated continuously by the DNA polymerase, which is called as the leading strand ones. Then the next, that is the parent strands of DNA, which runs to the 5-end to the 3-end opposite to the direction of the growing replication fork, and it is able to replicate discontinuously, is called as a lagging strand. Okay, now next in this we can say that Okazaki fragments, which is also known as it is a short sequences of DNA nucleotides which are synthesized discontinuously and we can say later linked together by the enzymes DNA ligase to create the lagging strand during the DNA replication one. Okay, so thank you very much. So do not spit in public place. Okay, thank you.